Hi to all of you. Welcome back to the series of lectures about the biosignaling. This is the eighth lecture of the series, and in the current lecture, we'll be discussing the steroid hormone signaling, the receptors to it, and the mechanism, taking example of the phosphoenol pyruvate carboxykinase as one of the cascading mechanisms of steroid hormone signaling. A brief introduction to the steroid hormone signaling, as we have already covered it in first two lectures of this series of the lectures. What happens for the steroid hormone signaling is that we know that steroid hormones control a wide variety of cellular functions, which include homeostasis, proliferation, differentiation of the cells, as well as apoptosis. And there are different kinds of steroid hormones which are secreted in the human body, which mediate the action via their receptors, which are called as steroid hormone receptors. And a particular thing about this steroid hormone receptors are that they are structurally intracellular proteins. They are not expressed on the surface of a cell, but they are present within the confines of the plasma membrane in the cytoplasm or in the nucleus of the cell, and they are crucial for the normal development and growth of their target organs and tissues, because they help in the downstream gene expression by activation from the steroid hormones after binding to them. A brief recapitulation from the previous two lectures of this biosignaling series. We know that steroid hormones belong to group 1 class of hormones, and they are lipophilic in nature. They are carried from the sites of the synthesis via bloodstream by different kinds of plasma proteins, which are serving as the carrier proteins for them, and then they enter the cell by diffusion because of their lipophilic nature. Inside the cell, they interact and bind to the specific steroid hormone receptors, which do the action in a very slow manner, and but the duration of action ranges from hours to days. For more information, you can visit the previous two lectures about the classification and signal transduction, lecture 1 and 2 of this series, to know more about the steroid hormones. As we know, steroid hormones are class 1 hormones, so their receptors are located within the cytoplasm. So the hormone has to move through the plasma membrane into this cytoplasm of the cell to interact with its specific receptor. So under basal conditions, what happens is that the receptors which serve for the steroid hormones are associated with heat shock proteins, and while they associate with the heat shock proteins, they remain inactive. Mostly the heat shock proteins with which they are 70, HSP 90, and 56, and these heat shock proteins behave as chaperones, which assist the inactive state of the receptors and keep them in the receptive cytoplasmic conformation until and unless the hormone binds to it. And these will become active only after hormone binds to it. So those which remain associated with the heat shock proteins are classically categorized as the type 1 steroid hormone receptors, and those which are rarely found are the type 2 SHRs, which are not needing HSPs to interact with them. So most of the abundant classes of the hormone receptors for the steroids are the ones which are associated usually under basal conditions with the heat shock proteins. As you can see in the left hand side of picture, these are classical type 1 steroid hormone receptors, which are usually associated with HSP 90 or HSP 70 or 56, and they remain inactive state in the inactive conformational state, but receptive to hormones. As soon as the hormone will bind to it, the inhibition by the heat shock proteins is lifted. So when they will become active by binding to the heat shock proteins, what happens is that the HSP 70 is or HSP 90 or whatever the case is released, and the hormone itself causes the conformational change in the receptor, and this receptor then binds to the second activated receptor to form a dimer, In this homodimer or as well as heterodimer, sometimes they are active and they move to the nucleus to bind to a specific region of the gene that are called as hormone response elements to cause the transcriptional activity. So this is the overview of the steroid hormone action, how they mediate their action. In an overview, what happens is that the hormone is first secreted in step 1, and they are associated with the proteins which carry the hormone to a target cell. Then the steroid hormones move inside as step 2. They bind to the receptor which is associated with the heat shock proteins. The heat shock proteins move out and the receptor undergoes conformational change, and it usually dimerizes, gets activated, and then this hormone receptor complex moves to the nucleus where it interacts with the specific target genes at a specific elements. Those are called as hormone response elements and result in the formation of more and more and expression of more and more mRNA, which in turn will cause the synthesis of proteins, usually the metabolic enzyme, which will mediate the biological function. I hope this is as clear. As we have seen along these lectures that the actual mediation of any kind of hormone signaling is actually done by the receptors. These receptors actually serve as the workhorses of the signaling. This is true also for the steroid hormone. But here the receptors are important in not only interacting with the hormone but also interacting with the DNA or the genome of the individual of the target cell also. So they need to be having a very controlled and regulated function. Essentially what happens for the steroid hormone receptors is that they are serving as the intracellular ligand activated transcriptional factors. So essentially they function in their active state as transcriptional factors. And the family of the steroid hormone receptors usually is divided into different types of domains. Here particularly for all steroid hormone receptors, they have 5 important functional domains. First one is the N-terminal domain, which is as you can see in the left hand side of the picture, marked as AB. Then there is an important one domain which is called as the DNA binding domain. This is the domain which interacts with the hormone receptor element or hormone response element of the DNA and causes its transcriptional activation, and it comprises of 2 zinc finger motifs within itself. Then there is a third highly variable hinge region, as you can see in the pink region of the diagram. And second important interacting domain in this hormone receptor is the LBD, ligand binding domain. This is the domain which binds with the hormone and interacts with it. So hormone will bind to the green region, ligand binding region, and then it will cause a conformational change, which in turn will cause dimerization as well as then activating DNA binding domain to interact with the hormone response element. And then there is a finally moderately conserved fifth domain, which is called as C-terminal domain. So N-terminal domain and C-terminal domain usually serve as an interaction for the dimerization, and hinge region is giving the flexibility for the conformational change, while as DNA binding and ligand binding domains are the domains which actually interact with something. DNA binding domain binds with the hormone receptor element or hormone response element after activation, while as ligand binding domain interacts not only with HSPs but also with the hormones for which they are meant to be relaying the signal. So the finality which is mediated through the hormone signaling is actually to bind these hormone receptors which are dedicated for each kind of an steroid hormone to bind to a specific target locations in the genes which need to be expressed. And the specificity of the steroid hormone is because of these hormone response elements. What are these? They are actually specific targets which are to be bound by the steroid hormone receptor after its activation, and they usually are the nucleotide stretches which are highly conserved and present in the regulatory part of these target genes. Usually they form a promoter region of the genes, of the metabolic genes, which need to be expressed at a higher concentration in response to these steroid hormones. And hormone response elements have been found to have a conserved sequence of two small 6 nucleotides, which are separated from each other by 3 base pairs spacer. As you can see in the estrogen response element, which is AGGTCA, and then the spacer of 3 base pairs, and then the palindrome also. Similarly, for the glucocorticoid response element, the palindrome flanking the 3 base pair on either side. The N, which is the 3 base pair, can be any variable nucleotide. So this response element is actually a part of promoter region of the target genes, which are to be under the influence of these steroid hormone receptors, and hence steroid hormone receptors will function as a transcriptional activators in presence of the hormone. When hormone is present, the transcription is meant to be at a higher pace. Another important aspect of these hormone receptor elements is that they are much more open to the crosstalk. That means that they are not only responsive to the steroid hormone receptors, but can be responsive to another signals as well through other receptors and through other effectors also. And most of the effect in the crosstalk is done by protein kinases, usually protein kinase A and MAP kinases, and their effective is CAMP, response element binding protein, which is also responsible for activating the promoter region and bind to the hormone response elements. As you can see in the left hand side of the right hand side of the picture, you will see that there is a crosstalk between other signalings as well. So the crosstalk is actually the convergence, which I have also covered in the previous lectures. You can see in the particularly CAMP signaling. One of the classical examples of the steroid hormone signaling is the phosphoenol pyruvate carboxykinase, which is a metabolic enzyme involved in gluconeogenesis, synthesizing glucose from the non-carbohydrate moieties, and it belongs to lyase family. Its expression is under the influence of a steroid hormone, glucocorticoid, particularly cortisol. But there is a crosstalk also via CREB under the influence of glucagon. So it functions during starved state, when we need to regulate the glucose homeostasis and synthesize glucose from non-carbohydrate moieties. It's found in two forms, cytosolic and mitochondrial. The cytosolic one is the one which is under the influence of glucocorticoids. So the transcription of the gene which codes this phosphoenol pyruvate carboxykinase is stimulated by glucagon, glucocorticoid, retinoic acid, as well as CAMP, and is inhibited by insulin, as we have already seen in the RTK signaling. Phosphoenol pyruvate carboxykinase is important and key enzyme of the gluconeogenetic pathway, because it converts the oxaloacetate into phosphoenol pyruvate and carbon dioxide. As you can see on the right hand side of the picture, it utilizes GTP moiety for it, for the conversion, and this is important because it helps in the bypassing the important reaction which is catalyzed by pyruvate kinase. So this reaction is antagonistic to the pyruvate kinase reaction. It supplies, it helps in the supply of the glucose from the non-carbohydrate moieties. So this enzyme needs to be in highly functional state and highly expressive state in starved state of a cell. So it is synthesized in liver in response to lower levels of blood glucose. When lower levels of blood glucose will happen, what happens is the glucocorticoids and the glucagon will be synthesized and released in the blood, which in turn will cause the affected cell to increase the expression of the phosphoenol pyruvate carboxykinase C form of gene. So the transcription will be activated and expression will be high under the influence of cortisol and glucagon. Particularly here, cortisol is the one which mediates its effect through the hormone receptors which move to the nucleus and cause the expression of this phosphoenol pyruvate carboxykinase gene, as well as via CREBP, which we know is under the influence of CAMP, and which in turn will function under the influence of glucagon. As I already had told you about the hormone response elements, here the hormone response element is particularly the glucocorticoid response element, which is located in the 5' untranslated region of this phosphoenol pyruvate carboxykinase gene, and has a sequence of GGTACA, and it is bound by the specific receptors, which are here glucocorticoid receptors in dimeric form. When dimers move to this region, they will cause the increased binding of the polymerase, and hence the increased expression of the phosphoenol pyruvate carboxykinase, resulting in the more and more mRNA, which in turn will be converted to enzyme by translation. So binding of this transcriptional factors, whether those are the glucocorticoid receptors or the CREBP, will result in the increased mRNA synthesis of this gene. So the overall signaling will be mediated by the decrease in the blood glucose concentrations, which will cause increased secretion of glucocorticoids, which will increase the hormone receptor complexes and cause the dimerization. Then this will move to the nucleus and will cause the increased transcription of the PEPCK gene, which will increase the synthesis of more and more mRNA of the particular PEPCK, which in turn will cause the high expression of the enzyme and lead to the increased conversion of pyruvate to glucose. So the net effect of the glucocorticoid signaling will be the overexpression of the PEPCK gene, which will affect the availability of the PEPCK enzyme, which in turn will affect the metabolism by converting more of a pyruvate, which is fed by the non-carbohydrate moieties, to phosphoenol pyruvate via oxaloacetate. First reaction catalyzed by pyruvate carboxylase, and the second is by the PEPCK under the influence of glucocorticoids. So this was it about the steroid hormone signaling. You can visit my Amazon page for the Kindle and paperback editions of different books on biochemistry and molecular biology. And do subscribe to my channel for further videos which I will upload in due time.